So today we are going to discuss muscle contraction. Okay. Now, when I am talking about muscle contraction, okay, actually we are going to discuss skeletal muscle contraction. Okay. Skeletal muscle contraction. Now, we have already discussed the structure of skeletal muscle and the proteins associated with this skeletal muscle, right? That is actin, myosin and all these things, okay? We have discussed A band, I band, okay? Now, today we are going to discuss the muscle contractile machineries, okay? Sorry, muscle contractile mechanisms, okay? Now listen, when I am, trying to teach the muscle contraction, actually I am trying to teach a very basic concept known by the name of excitation contraction coupling, okay? This is the basic concept of muscle contraction. Now listen, what do you mean by excitation contraction coupling? Now, point you need to understand is this, that excitation contraction coupling means whenever a nerve gets excited, contraction of a muscle occurs, and these two things, that is excitation of a neuron followed by contraction of a muscle, these two are coupled process. That means these two are occurring at a what? Single phase time. That means they are occurring at almost in a single phase, okay? That means they are occurring at regular intervals without any intervening delay, okay? That means suppose this is a muscle, okay, this is a muscle, okay, and this is a neuron. Suppose you already know that actually this is not the correct way to draw any neuron which is entering into a muscle, right? We have extrafusal fibers inside the muscle and interfusal fibers inside the muscle, right? We have alpha motor neuron. Suppose this is an alpha motor neuron. Suppose this is alpha motor neuron, okay? Suppose this is an alpha motor neuron, and whenever you excite this alpha motor neuron, you must understand this muscle spontaneously becomes excited and it contracts. Okay, it contracts. Okay, clear? That means whenever alpha motor neuron gets excited, then and there the muscle contracts. That's why excitation, contraction, these two things are coupled to each other. That means if excitation of alpha motor neurons doesn't occur, then contraction of muscle will never occur. These are coupled process. Okay. Now listen. The first mechanistic approach towards the muscle contractile theory was provided by two scientists known by the name of Huxley and Huxley. Okay, Huxley and Huxley. What they said is that whenever muscle contraction occurs, the Z lines comes closer. That means sarcomere of the muscle shortens. This is known by the name of sliding filament theory. Okay, now you need to understand this. Huxley and Huxley studied muscle contraction when there was no concept of genomes, when there was no concept of modern biotechnology or something like that. But now we know that using the concept of modern electron microscopy and all these things, we have now unraveled the each and every structure associated with this sliding filament theory. Now listen with all your attention. In sliding filament theory, what you will see is, suppose this is your Z line, okay, suppose this is your Z line, okay, suppose this is your Z line, okay, and these are your, suppose these are your Z lines, this is your A band, right? This is your A band, right? Now listen, when these Z lines come closer to each other, then we call it shortening of sarcomeres, right? This shortening of sarcomere was seen under microscope, and this mechanistic approach was named as sliding filament theory. That means what? Remember with all your attention. Sliding filament theory is actually one of the earliest attempt to explain excitation contraction coupling. Okay, but we now know that sliding filament theory is correct, it's true, but we now know that how this sliding filament theory actually works, because we now know this is what myosin, this is what myosin. Now listen with all your attention. We now know that how this actin and myosin, this is actin, right? How this actin and myosin interacts with each other, and how one myosin interacts with one actin, and how that myosin molecule produces a contractile motion. The basic molecular mechanism of this one myosin molecule is known as excitation contraction coupling. Okay, now listen. I have already stated at first that excitation contraction is a coupled process. That means either excitation will occur or contraction will occur, right? If you think this is right, then you are absolutely wrong, because excitation contraction is a coupled process. That means if excitation doesn't occur, contraction will never occur, and there is no contraction without excitation. Remember this. Suppose if you denervate or if you cut down alpha motor neuron, then there will be no contraction to this particular muscle. Okay, now listen with all your attention. You must now understand how excitation and contraction are coupled. This is the first thing you need to understand today. How excitation and contraction are coupled, this is the first thing you need to understand. Okay, now we will now be talking about excitation contraction coupling. Now listen, whenever I am talking about excitation contraction coupling, you must understand that is excitation contraction coupling is a continuous process as well as it's a coupled process. That means if excitation of alpha motor neuron doesn't occur, then contraction of a particular muscle will never occur, right? Now listen with all your attention. Suppose if I say that this is your, what is this? Suppose if I say this is your muscle, okay, and this is your alpha motor neuron, okay, this is the end foot of alpha motor neuron, okay. Now if this is the end foot of alpha motor neuron, you must understand this very basic thing. You should be clear in your mind that if you draw like this, if you draw any alpha motor neuron or any end foot of neuron like this, you will get a big zero in your exams. Why? The number one reason is this: every neuron ends in a muscle in a very much dramatic fashion, which is known as neuromuscular end. That means there is a difference between two neurons interacting with each other and a neuron and a muscle interacting with each other. There are basic differences, you know. These differences are very important for you to learn. Why? Because these differences are known as neuromuscular transmission. That means whenever a neuron interacts with a muscle, then ordinary synaptic transmission is actually not occurring. Here there is neuromuscular transmission of what? Synaptic vesicles or neurotransmitters. Then there is a particular change in the histology and physiology of end plate of alpha motor neurons. Okay, that means when any alpha motor neuron innervates a muscle, a significant change in the interactive state occurs. That means whenever two things come together, that is muscle and neuron, then normal interaction between two neurons doesn't occur. That means whenever there is an alpha motor neuron interacting with a muscle, then the end foot of the neuron enters the muscle cell, enters the cell membrane of muscle cell, and this is actually allowed by a special type of structure known as neuromuscular foot. This is called neuromuscular foot. Okay, now listen with all your attention. Whenever these neuromuscular foots are present, then there is a peculiar difference between a normal muscle cell and normal muscle cell membrane and a neuromuscular foot membrane. Whenever there is neuromuscular foot, these muscle membrane are different. Now listen. Can you tell me what is the similarity between a neuron and a muscle? What is the similarity between neuron and a muscle? A neuron can generate and propagate action potential, right? A neuron can generate and propagate action potential. A muscle cannot generate action potential, but it can propagate action potential. Why? Because muscles have enormous amount of sodium channel in them. Muscles have enormous amount of sodium channel in them, and for the generation of action potential, you need sodium channel, voltage gated sodium channels. But they lack few channels, that's why they cannot generate action potential. If they can generate action potential, what will happen? Then these muscles will never be under the control of neurons. They will automatically contract. You know, these things happen, don't worry, these things happen in a particular type of muscle cells known by the name of cardiac muscles, right? Cardiac muscle does the same thing. They don't have this, neurons which are actually controlling them. You know what is the peculiarity of heart muscle? What I am talking about is excitation contraction coupling, right? But if I say that heart muscle can generate its own rhythm, what do you mean by this? That means they don't need excitation. They don't need excitation? Wrong. They do need excitation, but these excitation are present within the cell itself. That means heart contains specialized cells which can generate action potential in situ at that point of time. That means they don't need external neurons to excite the muscle for contraction. You know who are these cells who actually are responsible for excitation of heart muscles? They are known as P cell or pacemaker cells. Synovial node is nothing but an aggregation of these P cells. Synovial node is nothing but aggregation of these P cells. Okay, that means you need to understand is this, that in skeletal muscle, alpha motor neurons are very important. Why? Because because skeletal muscle doesn't have these P cells. That's why they are very important. Now listen. If I draw this area here, then it will look like this. Okay, this is what? Muscle membrane. This is what? Muscle membrane. This is what? Alpha motor neuron, right? Now listen, I have already discussed that how synaptic transmission occurs. That means how I've already discussed how a particular neurotransmitter comes out of a synaptic vesicle from presynaptic neuron. I've already discussed how a particular neurotransmitter comes out of a presynaptic neuron, which is known by the name of synaptic transmission. And now listen, suppose acetylcholine. Now why acetylcholine is coming out? Now listen, acetylcholine is the preferred choice of neurotransmitter in a neuromuscular transmission. Acetylcholine is the preferred choice of neurotransmitter when there is a neuromuscular transmission. Okay, from a neuron, muscles are getting excited. That's why it is called neuromuscular transmission. Okay, that's why they are called neuromuscular transmission. Now listen, the most important thing you need to understand is this, that in a neuromuscular transmission, the most crucial part was release of acetylcholine. You already know this because in the synaptic transmission we have read about docking, priming and fusion. This is the basic molecular mechanism of neural things. Now what is muscular transmission? Now listen, this acetylcholine will bind with what? Now listen, acetylcholine has receptors in the cell membrane of muscle cells. Acetylcholine receptors are present in the cell membrane of muscle cells. Now what are these receptors? Now listen, acetylcholine have two types of receptor. One is cholinergic another is muscarinic. Okay. Now listen. Cholinergic receptors. Okay. Now listen. Why they are called cholinergic receptors? Because they are stimulated by acetylcholine. They are stimulated by acetylcholine. Right? Sorry. Nicotinic, sorry. Why I was wrong? Because, no, listen, why I was wrong? Because nicotinic receptors are present. Now listen, acetylcholine receptor has two types, that is nicotinic receptor and muscarinic receptor. You know how these receptors are named? Receptors are always named according to their agonists and antagonists. Cholinergic receptor cannot be a receptor of acetylcholine receptor because cholinergic receptor is another type of receptor. Listen, sorry. Now, nicotinic receptor, now listen, nicotinic receptors are present in the neuromuscular junction. This is called what? This is called neuromuscular junction. Okay, this is known by the name of neuromuscular junction. Clear? Now listen, important point you need to understand today is this, that in the neuromuscular junction, always, now listen with all your attention, always nicotinic acetylcholine receptors are present. We will have a detailed discussion on these acetylcholine receptors when we will be talking about pathology, as well as, when we talk about pharmacology. Why? Because there are many types of receptors, there are many types of drugs which are parasympatholytic, parasympathomimetic. Okay, that means we'll talk about this later. Okay, and those drugs actually bind with these muscarinic and nicotinic receptors. Now listen, what is the basic fundamental difference between nicotinic receptor and muscarinic receptor? You all know. Why you know? Because muscarinic receptors are fundamentally GPCRs, G protein coupled receptors. Okay, muscarinic receptors are fundamentally GPCRs, G protein coupled receptor. Tired of reading all these things, tired of reading physiology. And acetylcholine receptors are nothing but ligand binding, ligand gated ion channels. Nicotinic receptors are ligand binding ion channels. Now listen with all your attention. This is alpha, okay, this is beta, this is gamma, sorry, alpha, beta, gamma, delta, and in between beta and gamma there is alpha again. Okay, now listen, alpha, in between alpha and gamma there is a site for acetylcholine binding. Okay, in between alpha and gamma there is a site for acetylcholine binding, and between alpha and delta there is a site for acetylcholine binding. Okay, right? Now listen, suppose this is your what? This is your NM type nicotinic acetylcholine receptor, NM type nicotinic acetylcholine receptor. Now listen, when acetylcholine binds with the specified sites, then sodium enters the muscle, and with the entrance of sodium, action potential start to propagate. That means they have now started an action potential like potential differences which can travel. Why? Because action potential traveling depends on what? Sodium channel opening and closing. That's it. Clear? Now listen with all your attention. The peculiar thing you need to understand is this, that these action potentials or these potential fluctuations or these potential difference fluctuations travel in a membrane of muscle, but these muscle membranes are very well adapted to use this action potential as well. Why? Now listen with all your attention. Now listen with all your attention possible. Suppose this is one of the site which is now you need to understand is this, that suppose acetylcholine is come out of the presynaptic neuron and here is the arrival of action potential. Now listen, action potential means what? How to show an action potential travel. Listen, inside of the cell is what? Negative, outside is positive. If you want to show that this patch of the membrane has undergone action potential changes, then just reverse the charges. Okay. Now listen with all your attention. Important point you need to understand is this, that suppose this is a muscle cell, okay, this is a muscle cell, and suppose you know there were extrafusal fiber and interfusal fibers, extrafusal as well as interfusal fibers, right? There are extrafusal as well as interfusal fibers, we all know that, right? Now after taking out the what, these are perimysium, endomysium, after taking out all these things we have got what? Cylindrical protein like structures, inside which there were what? These striations, that is actin myosin band. Now listen, this is A band, I band, right? Now listen with all your attention. This is what? This is a muscle cell. Now listen, these muscle cell membrane have innervations. These muscle cell membranes have innervations, and these muscle cell innervations, these muscle cell membrane innervations are called T-tubules. Okay, they are known by the name of T-tubules. Okay, they are known by the name of T-tubules. Clear? They are known by the name of T-tubules. Now listen with all your attention. Now when these action potential changes travel towards the T-tubule, now there is a change in the T-tubule. Now listen, in the T-tubule there are these things. In the T-tubule there are these things. Okay, these things. Now listen, these are the blind ends of, now listen with all your attention, these are the blind ends of sarcoplasmic reticulum. What is sarcoplasmic reticulum? Listen, everything of muscle is designated by the name of sarco. That is, sarcoplasmic reticulum means what? Smooth endoplasmic reticulum of muscle cells are known by the name of a special type of categorized organelle, that is sarcoplasmic reticulum. Now listen, these are ends of what? Sarcoplasmic reticulum or smooth endoplasmic reticulum of muscle. Now listen, these are the blind ends, right? And this is what? This is nothing but a cistern like thing. Okay, or what is cistern? A bulging. Okay, now listen, this blind end together with this bulging thing is known by the name of L-tubule or cistern, terminal cistern. Okay, terminal cistern. Now listen with all your attention. When action potential will travel down this T-tubule, there is a disturbance in this terminal cistern. What is that? Now listen with all your attention. Very too important, and in most of the cases these proteins were unrecognized when we were studying actually excitation contraction coupling, but these two proteins which we'll now be discussing, they were the central to the understanding of how an action potential helps in muscle contraction. That means how an excitation is actually converted into contraction. Okay, now listen, Listen with all your attention. Okay, suppose these two proteins make a single unit. These two proteins are called ryanodine calcium release channels. Now listen with all your attention. These two proteins constitute a single channel, that is ryanodine calcium release channels. Okay, that means these are the guardian. That means sarcoplasmic reticulum are calcium store. They stores calcium. Why calciums are present inside the sarcoplasmic reticulum, or why calciums are present always inside the endoplasmic reticulum? In your GPCR class you have also read that in the GQ, when IP3 was actually was formed in the cytoplasm, then IP3 was actually binding to IP3 gated channels, and then calcium was coming out. Why calcium is stored inside the cell in a special chamber? Because calcium triggers, in most of the cases, apoptotic pathway as well. That means if you don't control the release of calcium inside the cell, then cells will die automatically. Now listen, these are the calcium release channel, right? Now calcium, that means in another word, calcium is nothing but a poison for cells. Now listen with all your attention. So if this is a poison, then this should be contained in a special place. But do you think these calcium release channels have that gate to actually stop calcium from coming out? But we do need these gates. These gates are also present. Now listen with all your attention. These gates are also present. These gates are also present, which are present here. Okay, which are present here. Now listen, this is a specialized sexy protein which is having no legs but a belly. Okay, now listen, that means this is a protein having two subunits and a connector subunit. Two subunit having a connector subunit. Okay, clear? Now listen, this protein is known as dihydropyridine receptors. Clear? These proteins are known as dihydropyridine receptors, or in simple short form they are known by the name of DHPRs. Now listen with all your attention. Probably you don't know, but suppose if there is a person in your family who is suffering from angina pectoris, or who is suffering from coronary artery diseases, and if you consult any doctor, doctors probably have given a blocker of this particular channel. A doctor obviously has given a blocker of this particular channel, but those blockers are only selective for dihydropyridine receptors present inside your heart. You know what is the name of those blockers? The name of the drug is amlodipine. Amlodipines are dihydropyridine receptor blockers which are present only in heart. That means heart homologue of dihydropyridine receptor blockers are amlodipine. So this is amlodipine. Okay, so what is the function of amlodipine? Amlodipine actually reduces the force of contraction in heart. That means it reduces the force of contraction in heart. That means it reduces the contractility of heart, which is one of the major symbol for cardiac output. That means it reduces cardiac output to some extent in what? In those patients who cannot take care of their contractility by their own, because contractility depends on many factors, and if you cannot control those factors, then probably you are going to have an heart attack. Now listen with all your attention. So this is dihydropyridine receptor, okay? It's an L-type calcium channel release, it's an L-type, sorry, ryanodine calcium release channel is an L-type calcium channel, okay? Dihydropyridine receptor has two subunits. Now listen with all your attention. You must understand a very basic thing about dihydropyridine receptor now. Listen. Suppose this is another dihydropyridine receptor, okay? Suppose this is another dihydropyridine receptor. Okay, this is another dihydropyridine receptor. Now listen with all your attention. Suppose this is dihydropyridine receptor, this is dihydropyridine receptor. Now listen, these dihydropyridine receptors are present throughout this T tubule. That means they are present also here. Okay, I'm not drawing. Now listen, there's an important point you need to understand right now. That is, I have already told that dihydropyridine receptors have two domains, one which is attached with the T tubule and one which is attached with the ryanodine calcium release channel. Now listen with all your attention. That is, when action potential is coming down into the T tubule, that means what? Suppose there was calcium, there was action potential, and it is coming now here. After this patch of membrane is excited or depolarized, then this particular position will come when this will occur. Now listen with all your attention possible. Now listen. Suppose this patch of membrane have now become depolarized. But do you think this patch is now depolarized or polarized? Polarized. But there is a contrasting thing, you know, that a battery is formed here across the dihydropyridine receptors. That means there exists an electric field. And listen with all your attention. This is the very basic thing you need to understand. When a protein responds to any electric field, and if the electric field is going throughout its body, then they are known by the name of voltage gated channels or voltage gated proteins. If any protein respond to this type of electric field, okay, going throughout their body, then they are called voltage gated proteins. Okay, that means this particular segment of DHPR is a voltage gated protein. Now listen, that means with the arrival of action potential or with the activation of this muscle membrane, the outer domain of DHPR becomes excited because it's a voltage gated protein, and it will come towards this side. It will come towards this side. That means what? If this whole thing is coming towards this side, that means this whole thing is coming toward this side, means this whole thing will now shift, right? Okay, this whole thing will now shift, and calcium will now start leaking from the ryanodine calcium release channel. Now listen with all your attention. You should never forget that why these channel and why this receptor has been named as dihydropyridine receptor and ryanodine receptor. Now listen, ryanodine is a drug which is a poison. Ryanodine is a very good poison for you people. Why? Because ryanodine, this protein actually, or this compound actually, ryanodine or this compound actually blocks this channel, and dihydropyridine is another compound which blocks this channel or blocks this protein. That means now listen, it's a very important thing for pharmacology. Now listen, acetylcholine receptors are named according to their agonists. Agonist means what? Agonist means what? Those who help in a process. That means nicotine help in what? Acetylcholine transmission, and muscarine also helps in acetylcholine transmission. Now listen with all your attention. But dihydropyridine Dihydropyridine receptors and ryanodine calcium release channels are named according to their antagonists. That's a very important thing you have to understand. Clear? Now listen. Actually what happens when you take amlodipine is this. Amlodipine acts here and they block this calcium and they block this area, and even if action potential invade this particular domain, they will not be able to move here. That's why amlodipine is a what? Amlodipine is a anti-contractile drug. That means it reduces the contractility of heart. That means what? Why it reduces the contractility of heart? Because there is no calcium release, and calcium is extremely important for contraction of any muscle, remember this, okay? Also for smooth muscle, cardiac muscle, as well as skeletal muscle, okay? Now, so we have now discussed that how action potential invasion leads to calcium release. We have now discussed up to now that how action potential invasion leads to calcium release in that T tubule. Now listen with all your attention. This is T tubule. This is what? Terminal cisternae, okay? And here is calcium release channel, and here is what? Dihydropyridine receptor. This whole thing, this whole unit is called triad. This is triad, okay? Clear? This is called triad, okay? Now we are going to discuss that how this calcium affects contraction. That means we have finished the first portion of the excitation contraction coupling theory, that is the excitation. Now the particular patch of membrane is excited, and we have discussed how this patch of membrane travels. Now we are going to discuss is how this calcium release leads to contraction of muscle. Now I am not going to discuss a significant portion of this theory. Why? Because we have already discussed it in the previous class. That is, we have already discussed that how calcium binds with TNC, troponin C subunit, and how with the binding of troponin C, troponin I shifts its position, and active site of actin is what? Exposed. Active site of actin is exposed, and with the exposure of active site of actin, with the, are you lost? Thank God. With the exposure of active site of actin, okay, myosin interacts. Now listen, now listen, to explain sliding filament theory, you should always remember, many people, when I actually take classes, okay, there are many people who are confused. What is sliding filament theory? What is excitation contraction coupling? What is cross bridge cycle? Now listen with all your attention. These 3 points are very important for you to understand because they are correlated. Listen. To explain sliding filament, excitation contraction coupling theory was given. To explain the contraction part, cross bridge cycle concept was given. Clear? Clear? Okay? Thank God. Okay. So to explain sliding filament, excitation contraction coupling theory was given. To explain the contraction part of the excitation contraction coupling theory, cross bridge cycle was given, that how this contraction is occurring after the release of calcium. And we have already discussed a significant portion, that is, this is your what? G-actin, right? And this G-actin forms F-actin, right? Clear? Okay. Now listen. So this is your F-actin, right? Or G-actin. These are G-actin, right? This whole thing is called F-actin. Now listen. This was your myosin. Yes. Now listen. This was what? Heavy myosin, heavy meromyosin, and this is light meromyosin. Now listen. Now there is a problem here. Why? Now we are going to some extent details. Now listen. In the heavy meramycin part, there are two special portions. One is this, another one is this. Now listen. Why these two portions are special, you know? Because these two portions actually move during contraction. That's why specialized amino acids are present here. Clear? That's why specialized amino acids are present here. Clear? Now listen. Suppose this is your tropomyosin, okay? This is your tropomyosin. I've already shown what is tropomyosin. Now, this is what? This is troponin T, TNT, right? Why it is troponin T? Because this part of the troponin is connected to tropomyosin. This is what? Okay. This is what? Which is covering the active site of actin? TNI, inhibitory. And which is, what is the name of this site? What is the name of this site? TNC. Clear? Now listen. The calcium, now listen. Suppose this is your T tubule, okay? And from here calcium is coming, and you know why, you know how and why, right? So now calcium will bind here. Calcium will bind here, and due to the binding of this calcium, what will happen? There is a shift. That means this will come here and this will go here. And you know what was here? This is suppose T and T, and you know what was here? There was already what? Myosin. Okay. Now listen. When this troponin I inhibitory site has moved on, then this particular portion of myosin will interact with actin, right? Active site of actin. Now listen with all your attention. Actually, we'll discuss it in great details now, that how this myosin interacts with actin. Okay. Okay. So now we'll be discussing cross bridge cycle. Now listen. Cross bridge cycle starts with this basic thing that whenever actin and myosin interacts, okay, suppose this is your actin, F actin, okay. Now listen, this is 1, 2, 3, 4, 5, 6, 7. Now listen, important point you need to understand is this. Suppose this is myosin molecule, okay. Now in a cross bridge, at the end of cross bridge cycle, what will happen is the head of this myosin will be attached to 3. That's it. Here is your Z line, okay, this is Z line. So correspondingly here also there is Z line, and that means whenever cross bridge cycle is occurring, that means these two sarcomeres are coming together. That means this actin will approach the central area, right? That is the cross bridge, that is contraction, that is shortening of sarcomere, that is the definition of contraction. Now listen. So what we are going to achieve is this, that at the end of this cross bridge cycle, this actin head, sorry, this myosin head will be attached with the third actin, okay. Now listen with all your attention. To accomplish this, we need several steps which occur at a nanosecond speed. Yes. Now listen. At the starting point of cross bridge cycle, what happens is myosin head is attached with actin, okay, because there is no inhibition, which has been removed by calcium, okay. Now listen. When ATP binds with this myosin, what happens is, now listen, if you consider the structure of myosin head, it looks more or less like this. That means there are two sides. One is actin binding site, okay, another is ATP binding site. Okay, actin binding site, and another is ATP binding site. Now listen, when ATP binds with the ATP binding site of myosin, ATP binding site of myosin, then what happens is suppose this is the, suppose what happens now is this myosin molecule is now not at all attached with the actin molecule. That means ATP is required for the detachment of actin and myosin. Now listen with all your attention. When you are dead or in a corpse or in a dead body, this ATP is not present. That means there is a complete what? Complete stiffness in the dead body, because these muscles will never again occur at its normal position. That's why this is called rigor mortis, stiffening of muscles after death. Okay, here listen, the dead body of corpse or a particular hand muscle or leg muscle may move, but it doesn't mean that this whole corpse will move. Why? Because it is nothing but tightening of muscles. Now listen, after certain period of time, these proteins will degrade, because all of the proteins which are constituent of our human body have a half-life cycle, half-life period, and when these proteins will degrade, then total smoothness in the body of the corpse will occur. Okay, but it will occur only if you give subsequent time. Okay, if you give many time, if you leave the dead body for at least 24 hours, then that will occur. But within 10 to 12 hours, rigor mortis set. Rigor mortis actually is occurring. Now listen, when ATP binds with the ATP binding site of myosin, then what happens is the actin myosin bridge is broken. Okay, now ATP is bound to myosin ATP binding site. Now listen with all your attention. At that point of time, what will happen is from this ATP, one phosphate will come out, and this ATP will be converted into ADP. Now when this occurs, now listen with all your attention. When this occurs, there is an important point you need to understand is this. When this occurs, so now suppose this is 1, 2, 3, 4, 5, 6. Now listen, suppose if ATP is now attached with this head, then actin and myosin bonds are broken. Now listen with all your attention. With the hydrolysis of ATP, ADP and inorganic phosphate is produced. Now with the release of this inorganic phosphate, there is an angular change in this myosin, and this myosin will form a collinear structure. That means it will be parallel to other myosin as well. Okay, now listen with all your attention. That means this whole structure will now be like this: ADP PI. Okay, clear? That means if I redraw this structure only, then it will be like this: ADP PI only. Okay, now listen, after some times what will happen is, just after few nanoseconds, what will happen is, listen with all your attention. After few nanoseconds, what will happen is, Clear? After a few seconds, what will happen is this myosin will again go up and catch the third actin, okay, in place. This is called weak Cox state. This is called weak Cox state. Okay? Yes, that's not horny. Okay. Now listen. When this weak Cox state will occur, then weak Cox state is quickly followed by a strong Cox state or a strong covalent state. That means in this strong Cox state, this phosphate comes out, and actually the proper stroke of this myosin on this actin will occur now. That means whenever this inorganic phosphate will come out, okay, whenever this inorganic phosphate will come out, then a strong conformational changes induces a stronger covalent bonding between actin and myosin, and a conformational change in the myosin occurs, due to which this myosin molecule will now move towards the H band, okay. That means they are moving towards the H band, means this is Z line and here is Z line, here is again what, actin, and this is the H band. They will move towards the H band, and due to this contraction, they will now actually move the actin molecule from 3 to 2. That means they were touching the 4 in the first place. In the second place, they are now detached due to the attachment of ATP in the ATP binding state, ATP binding site. Then after the hydrolysis of ATP into ADP and PI, what will happen is this myosin head will get attached with the third actin molecule. Now listen, when this is occurring, this is called Cox state. That means there is already a covalent interaction between this actin and myosin, okay. Now when this phosphate comes out, what happens is, this covalent interaction between the third actin and the myosin molecule leads to the contraction, due to the removal of phosphate, the contraction or conformational changes induced by the removal of phosphate actually causes a change in the angle towards the H band, and due to this a movement occurs, due to which the Z lines of the two sarcomeres come closer. Now listen, the last point you need to understand is this: you will have to remove this ADP as well. And this is the last state, that is, in the last state this ADP has been removed, and this myosin is now attached with third molecule. And now listen with all your attention possible. That is, due to the removal of ADP from this myosin molecule, we are again starting this whole process in a similar fashion, but when we are starting again this process, then instead of interacting with the fourth actin, this myosin head is now interacting with the third actin. That means due to this whole process, which occurs in a nanosecond fraction, okay, this particular actin line has gone here. That means it is approaching the age zone, age band. Now listen, this is called cross bridge cycle. Why it is called cross bridge cycle? Now the bridge means what? Bridging between actin and myosin, and cross means what? Crossover between ATP and ADP sites, or crossover between ATP and ADP. That means, listen with all your attention, if there is an interaction between actin and myosin, there is an interaction between actin and myosin, who will have to come to detach them? ATP. Now listen, for the contraction, for the proper contraction, who is responsible? The outgoing of phosphate. Yes, outgoing of phosphate. Okay. Now listen, for the super strong COX state, outgoing of phosphate. Now listen, for the starting of cross bridge cycle, who is responsible? For the starting of cross bridge cycle, ATP entrance is most important in the ATP interaction in the ATP binding site of myosin is the most important factor. Okay. Now in dead body or corpse, this ATP is absent. Therefore, all of these muscles or in your body muscles actually cannot contract because they are in COX state with the actin. Okay. Now listen with all your attention. So all these things have started because calcium was present in your cytoplasm. But I have already told that calcium is very much poisonous with respect to what? That they can actually activate many enzymes which are not at all what? Wanted by the cell at high concentrations. Okay, because you have already learned in GQ signaling that calcium binds with calmodulin and they activate CAM kinases, and CAM kinase can activate many proteins. Okay, now listen, to stop the skeletal muscle contraction, we need sequestration of calcium or reuptake of calcium or restoration of calcium into the sarcoplasmic reticulum. For this, we need a protein known by the name of calsequestrin. What is this? Now listen, probably you have heard of this, which is called cal. Cal means calcium. What you will do? You will sequester the calcium. What is sequester? Withdrawal. You will have to withdraw the calcium from the cytoplasm. Sequestering means withdrawal. That means calsequestrin. In means protein. This protein withdraws calcium from the cytoplasm. Now listen, we will be talking about this particular protein now. Listen, what is the importance of calsequestrin? Suppose this is sarcoplasmic reticulum and this is suppose calsequestrin. What it is doing? It is taking in calcium from the, suppose this is your cell, muscle cell, and it is taking calcium from the cytoplasm, and this is suppose calsequestrin. Okay. Now important point you need to understand is this: calsequestrin is an APT driven protein channel. That means it is an active channel. Okay. That means it requires ATP to form ADP plus PI. Okay. That means what? Listen, for the relaxation of muscle you need what? ATP. For the contraction of muscle you need ATP. Now listen, in your tough exams you will not get this type of question that why it is important, why ATP is important. No, what you will get is in which state of contraction ATP is required, in the cross bridge cycle, or in the which part of the cross bridge cycle? For detaching the actin and myosin. That's it. For detaching the actin and myosin only you need ATP. After that, for the cross bridge activation and cross bridge contraction, you don't need ATP, you need ADP and phosphate, right? Now listen, and in and in skeletal muscle relaxation, where you need the ATP? You need the ATP for the activation of calsequestrin. Okay. Now, how this calsequestrin gets activated? When there is a certain there is a certain amount of calcium present in the cytoplasm, then this calcium actually activates this calsequestrin. Okay. Now listen with all your attention. There are many proteins which actually work like calsequestrin. Okay. That means in the in the skeletal muscle contraction as well as in the skeletal muscle relaxation, you need ATP, and you now know where do you need those ATP. Okay. So we have discussed what is sliding filament theory, not in details obviously, because the details of this siding pyramid theory is nothing but an important investigation. We need an important investigation of what? Excitation contraction coupling. We have done that. How muscle cells get excited and how this contraction is coupled. Next, we have also discussed what is relaxation, how this relaxation occurs. Withdrawal of calcium or sequestration of calcium is the most important thing in relaxation. Clear? And how it is done? By calsequestrin protein, which is present in the smooth endoplasmic reticulum. Okay, thank you.